Okay, can everyone hear me? I'm going to assume yes, since everybody's typing now. Okay, so my project is working on drought-resistant wheat, which is conferred by a translocation of the 1RS arm from rye. And I work at Jorge Dubcovsky's lab here at UC Davis, same lab as Brittany. So a little bit of background. Wheat breeders have used. Hold on. Can you turn off your mic? I can hear myself talking. Okay. So wheat breeders have used the rye chromosome, or have used rye as a genetic resource for wheat for a long time. Rye is a very hardy crop. It can grow under a lot of different stressful conditions that wheat has a tough time with, and basically the goal is trying to combine those that hardiness of rye with the high yield and the high quality that the wheat has. Specifically from rye, the short arm of rye chromosome 1 is really gene rich, and it contains a lot of genes for pest and disease resistance. That's the one that I'm using in my research. It's the short arm is just called 1RS. So translocations involving this chromosome arm, 1RS, they have a lot of different traits with them. There's all kinds of different disease resistance traits, pest resistance traits, but it also has some bad traits associated with it, which is kind of limited its usefulness in breeding programs because the wheat isn't as high of quality, so you can't use it for as many things. Adam Lukaszewski down at UC Riverside here in California, he addressed this problem back in 2000, I believe, when he engineered a recombinant chromosome that contained a 1RS translocation onto the 1B chromosome of wheat, and he engineered it so they had two interstitial segments of wheat chromatin on that rye arm, which corresponded to regions that were associated with the poor dough-making characteristics that are associated with the 1RS chromosome. So he identified these two regions as the SEC1 locus, which is a rye gene. It encodes for secalene proteins, which are rye storage proteins. And they reduce dough quality, they reduce the mixing strength. And he also identified another locus, the GLB1, GLB3 locus. And that is a wheat-associated gene, which encodes for gliadins and glutenins, which basically increase the dough mixing strength. And so he's basically trying to get rid of something on the rye chromosome, and he's trying to introduce something else in a different place. Here's just kind of an overview of what the chromosomes look like. So you can see at the top we have the full 1RS chromosome, and I just have the red is rye chromatin and the green is wheat chromatin. So what he started with was at the top, and what he ended with. Yeah.
We're still in the background.
Oh, really?
I'm wondering if I can just skip in the head.
Did that work at all?
Okay, what's the title of the slide we should be on?
It should say background, but it should be kind of a picture.
Okay, of two chromosomes?
Yeah.
Okay, we're in the right spot.
Okay. Alright. So, yeah, basically this one at the top is the whole rye chromosome, if everybody's on that same one. People are still typing. Okay, I can try loading this again. That'll help. Yeah, or if you guys just want to change it yourselves, I can tell you when I change slides. That might be easier. Oh, now I'm back at the beginning.
We lost the control. Yeah, we're on the right side. Oh, lost it.
Okay. I don't know what's happening, but let me try and read it again. Okay.
There
Can everyone see it now? Okay. So, as I was saying, on the top here, you can see we've got the centromere, and then this is looking at just the short arm of chromosome 1b, which has been translocated with the short arm of chromosome 1 from Y. So the red color corresponds to Y chromatin, and the green color corresponds to weak chromatin. So on the top is basically the full 1RS translocation, and the bottom one is this chromosome that Adam Drought engineered, which has 2 pieces of weak chromatin at the Sec1 and the GluB1, GluB3 locus. So this is just kind of where I came in for my project. So when we were growing these 2 different types of, or these 2 different genotypes in the field, they were isogenic aside from that one chromosome. One had the full 1RS translocation, one had that recombinant chromosome. And we noticed that under conditions of water stress, the full 1RS chromosome had a much higher yield than the MA chromosome, that engineered one that had the 2 interstitial pieces of weak chromatin on it. And we also noticed that the effect of the yield differences seems to be dependent upon the genetic background. So all the initial work by Adam was done in the Pavlone background. But we moved both the full 1RS and the 1RS MA chromosomes into 2 different backgrounds called Han and Attila, and they're basically just more sensitive to water stress than Pavlone was. So we can see the differences a lot more easily here. So it's a lot easier for us to get a phenotype and work with these chromosomes in these backgrounds. So now that we know something's going on, my project goals are, first of all, to establish clear phenotype to facilitate the selection of varieties, because we can tell through yield, but we have to wait an entire season, and it would be ideal if we could choose these traits a little bit earlier so that we can actually cross things before they go to seed and all that. We can tell, basically just at an earlier time point, which ones are expressing the drought tolerance. So once I nail down a phenotype, I need to determine which of those 2 wheat regions, either the Sec1 or the GluB1, GluB3 locus, is responsible for the differences in yield. And if I do determine that it's only one of those regions, I like to find map that region and eventually clone the gene or genes that are responsible for this field effect that we're seeing. So here's a little bit of my results from this past field season. Let's see if I can get a pointer here. So over on the left side, so we have our 2 different varieties. These left 2 graphs are the Attila variety, and these right 2 are the Han variety. And everything that we've done so far, we've done several yield trials, and we always see a bigger difference in the Han varieties than we do in the Attila varieties between the 1RS and the MA chromosomes. So whatever it is that's associated with this chromosome, it is dependent upon genetic background, and people have seen that over and over and over again for all kinds of different drought stress traits. So as you can see, we have over here the red bars are the water indices, well, this is just one water index. This is the normalized water index 3, and these measurements were taken with our JAZZ unit. So this is basically what we're using it for, is to kind of try and nail down the water status of the plants. So this red bar is the water status of the plants in the dry trial, and the blue bar is the water status of the plants in our irrigated trial. We had 10 blocks of each genotype and each variety repped 2 times. So we had 20 blocks for each variety and each genotype in each trial. So there were 40 blocks total for Attila 1RS, 40 for recombinant, 40 for the Han 1RS, and 40 for the Han recombinant. Basically, we can see here that there's not much of a difference in the Attila 1RS between the dry and the watered. They're about the same. There's a little bit of a difference maybe between the 2 different trials in the 1RS recombinant chromosome and the full 1RS translocation, but it wasn't a statistically significant difference when we ran this through SAS. Do we already have drought-resistant wheat? I'm not sure what exactly you mean by that. I mean, there's all kinds of different drought resistance. I think we're just kind of chasing after this one gene because we found this already. I assume you're asking. So we're thinking that our 1RS wheat is drought resistant. So that's why we're expecting that there probably shouldn't be too much of a difference between the irrigated and the dry. But if you look over here at our Han 1RS recombinant chromosome, there is a significantly lower yield in the dry plants than there is in the irrigated plants. Yeah, so that's kind of what. So Pavone and Attila seem to be more drought resistant than Han is, which is why we moved it out of the Pavone background and we moved it into these 2 different backgrounds because they were a little bit more sensitive to drought, so we can see the phenotype easier. I believe there was still a difference in Pavone. It just wasn't as big of a difference as we're seeing in Han, for example. So it's just a lot easier to work in this background. So yeah, as you can see with the full 1RS translocation in Han, there's not much of a difference. They look pretty much the same. But with the recombinant chromosome, there's a much higher yield in the irrigated trial than there was in the dry trial. So we're definitely seeing something that's drought related here. And if we look at my next slide, so here's some pictures of the plants. I don't know how easy it is to see these, but basically what we're trying to do with our jet spectrophotometer is telling us basically what we can see with our eyes. It's a little bit better because it is actually looking at the water status of the plant. But even just going out at the field and looking at these plants, you can see definite differences. So it looks like my slide got a little messed up from putting it on here. But on the left over here, we have the Attila plants. So this top left one here is Attila with the full 1RS translocation. And this bottom left one here is Attila with the 1RS MA translocation. So that's the recombinant chromosome. And you can kind of see on here that this top one up here maybe looks a little bit better than this one down here, but there's not a huge difference. And similarly, there's not a huge difference between this top left one, which is the Attila 1RS, and this top right one, which is the Han 1RS. So this is the same genotype in 2 different genetic backgrounds. And this is all in the drought-stressed plots. So these haven't been watered. But then when we look down here at this bottom right one, this is the Han 1RS MA genotype, and you can see that a lot of the leaves are kind of drying up, they're dead, and it's just all the ones that are still green, they're very curled. They're really showing the drought stress here. These Attila ones with the 1RS chromosome, a lot of the leaves are curled. It's a little hard to see in this picture, but the leaves are curling, but on these top two with the full 1S chromosome, the leaves are still they're nice and flat and broad. I think you can kind of see it on these. But we are definitely seeing differences in how the plants are responding to this water stress. So for a yield trial, we saw kind of what we expected. We saw, well, actually we were expecting to see about the same yield in the 1RS plants between the watered and the dry trials. There was a bit of a yield difference. We're still trying to figure out if this could be because we had a very, very wet winter and wet spring here, so we're not sure if that kind of messed with our drought conditions a little bit, since there was a little bit of a drought, but then it kept raining. So, but we did see that our HA and MA, the recombinant chromosome, it did have a bigger percentage difference in yield between the watered and the dry trials. And we also did 1,000 kernel weight, and this gave us more or less the same information that we've been seeing before. And that is to say that there was a higher 1,000 kernel weight in the water trials than there were in the dry trials. And there was more of a difference between the two trials in the HA and MA than there was in the Attila MA background. Okay, so what am I doing now? So since this past field season, I've been trying to get some genetic work done on determining the region of interest. So I want to find out which of these two regions is responsible for these differences that we're seeing. So what I need to do that is to perform canopy spectral reflectance with our JAZZ unit on 4 different genotypes. So I want to look at the full 1RS genotype, and I'm looking in both the Attila and Han backgrounds, but I'm focused mainly on Han right now because it shows just a much bigger difference. So I want to look at the full 1RS genotype, the full 1RS MA genotype, which has both of those wheat segments. And then I want to look at a genotype that has wheat only at the GLU1, GLUB3 locus and still has rye at the SEC1 locus. And then I want to look at the opposite case where the wheat segment's only at the SEC1 locus and it's rye at the GLUB1, GLUB3 locus. So for doing this, we had planted out 100 F2 progeny that were from a cross of a full 1RS with a 1RS MA chromosome. And I basically genotyped all of those. I had some issues with the markers. I could only get 2 of them to work. So I had to end up doing some progeny testing because these were dominant markers. So I could tell if they had at least one rye allele at the SEC1 locus and at least one wheat allele at the wheat locus, or at least one wheat allele at the GLUB1, GLUB3 locus. But I couldn't tell if those were heterozygous or homozygous. So I had to do progeny testing, and I planted out 24 plants from some selections that I made, and I did actually end up getting both of the critical recombinants I needed. I only got one of each, but that should be good enough. So my next steps are I need to plan out these recombinant progeny in the field season and perform that CSR on them to determine which of these 2 regions is responsible for the increased yield. I need to confirm the role in drought stress just because we're seeing some weird stuff. As you saw, the CSR that I did earlier in the season didn't correspond perfectly with the yield data that we're seeing at the end of the season. And basically we just need to confirm that this is drought related and not just a basic yield trait. Based on how the plants were looking earlier in the season, it definitely looked like there were differences in the water stress of the plants. So it does seem like it's probably drought associated, but we'd like to confirm that. And after I do determine which of these regions is responsible for the differences in yield, I need to develop markers to fine map that, and then start identifying my candidate genes. And if possible, it'd be great to identify the mechanism of what these genes are doing, and eventually make this available for wheat breeders so that we can have more drought-tolerant wheat. So acknowledgements, obviously I have to say thanks to Jorge Dubcovsky for all his help and doing all the past stuff and being my PI. Josh Haggerty helped me out with a lot of this stuff. He's been helping me run the statistical analysis. He went down to Imperial Valley where we had some wheat planted out and did the CSR down there. Yago Lowe, he kind of started this project a while back and ran some of the initial trials. Lucho was a visiting scientist we had here, and he helped me with some of the market development. And Guy Fukumoto, Kyle, Sharon, there's some students we have in the lab here that helped me out with some of this stuff. And I'd like to thank my funding, so the TCAP program and USDA and NIFA, obviously, and also from UC Davis, the Henry Jastrow Graduate Research Scholarship Award. So I think that's about it, if there are any questions.
Tyson, so we've been using the jazz out in the field, and I'm just wondering if you had any similar experiences to what we're having. The machine is overheating on us and freezing up, and we're trying to work with the company to work out these issues. But I'm wondering if you had any experiences like that.
So we didn't have any problems with the overheating. I have heard there was one other group that was having that problem, and basically the company just told them to wrap the unit in like plastic wrap and put an ice pack in with it, which doesn't seem like the greatest fix, but I guess it works. We did have a little bit of an issue with it freezing at first, but I think what this is due to is it's really easy when it's in the jazz pack, that little jacket that it has. When it's zipped all the way up, it's really easy to accidentally eject the SD card, and sometimes all it will do is you just won't be able to save things anymore, but sometimes it'll also freeze it, and it takes a little while to get it going again. Josh had some problems with this when he was down in Imperial. He said there were a couple times where it just completely stopped working, and he had to take the SD card out, download everything onto his computer from it, wipe it, and then put it back into the unit, and it started working after that. So I haven't had any problems with it personally, other than I think one time it froze, but I checked and it was because the SD card got ejected. So yeah, I guess that's something to look out for. Any other questions? Oh, how long did it take to take the reading? I think I had 96 different plots, and I think it was taking about an hour to 12 plots. So it's about, it was a whole day to do the 96 plots. Yeah, about 8 hours. I think I split it up over 2 days. So we did half of them one day and then half of them the next day. So Leanna's asking if heat can balance our drought stress phenotypes. Do you mean, is it maybe a heat stress phenotype rather than a drought stress phenotype, or is the heat kind of messing with the phenotype that we're getting from the jazz? Okay, so do we know that we're looking at drought stress and not heat stress? I think it is drought stress because when those pictures that I showed, when we're seeing the visual differences, it wasn't very hot in Davis. It was back in the spring. It's hot here now, but before the weather really started ramping up and getting really hot, we were having a good drought trial before the rain started coming. It wasn't too hot or anything. It was starting to get dry. So I don't think that the plants shouldn't have been too heat stressed. And we were seeing differences between the water plants and the not water plants. So I guess the water availability for the water plants would be better, so that could help them with cooling. But it does seem, it's kind of hard to start teasing apart heat stress and drought stress at that point when they're getting heat stress because they have less access to water. So it does seem like our plants probably do have more access to water. We've also seen this in some of our past trials. We've done carbon isotope discrimination, which basically tells you how much photosynthesis the plants are doing, and it does seem like the full 1RS plants are continuing photosynthesis longer than the other plants with the recombinant chromosome. So it does seem like it's a drought stress related phenotype. Okay, so Irina, I'm sorry, I don't know how to say your name, but she's asking about getting more readings from the jazz than you push the button for. We did have that problem. I talked to Jada about it, and she said that it's a bug with the way it was programmed. So basically, if you set up both of the channels, so we usually set up our downwelling with an integration time of about 10 milliseconds and our upwelling with an integration time of about 30 milliseconds. But every time we save a reading from our plants when we're taking the upwelling measurements, it also saves a reading from our downwelling channel at the same integration time. So basically, if you're trying to take readings with two channels, you end up with half of the files that got saved are junk because they're either underexposed or overexposed because they were taken with the wrong integration time and you weren't trying to take those readings in the first place. If you guys are using the Jazz units, we do have a Perl script that we use to parse everything. So basically, you can just cover up the detector whenever you're not trying to take a reading with that, and then at the end of the day, you bring it all back on your computer, and we have a renamer that'll rename all of the files in a single folder. and you run the parser, and it'll basically average together everything in between those blank measurements. So if you have 3 measurements of your plot, and then you have then you cover the detector and you take downwelling readings, and then you take a blank for the next plot. So you have 4 blank readings in there, and then you start taking your upwelling readings for the next plot. It will just treat all of those blanks as a single blank, so you don't have to worry about confusing yourself and accidentally averaging in some 0 values or anything like that. So if I haven't sent that out yet, I will soon. I've been really busy with trying to get my recombinants and stuff, but we do have some of that, and we've also been working. Actually, one of the students, Jun Lee, he's got a program for using the JSL. He found a way to get around the problem that we were having with it overwriting files every time you restarted it. So he's using a 3-channel jazz right now. I'm adapting it for a 2-channel, and then I'm going to send that out to everybody as soon as it's done. I did not get a chance to determine the length of the grain fill for these plants. It was my first year joining this lab, and the plants were already in the field, and I just had so much to catch up on that I didn't get a chance to make it out there very often. I only got out there for 2 different days for the jazz readings. I'd like to get the readings at a bunch of different time points throughout the year, but this year was a little rushed, so I didn't get as much data as I would have liked. Is there a way we can make this chat window bigger? It's kind of hard for me to see it on the screen. Oh, okay. So Jamie says she wonders if the length of grain fill would be easier to get than the CSR and might be more meaningful. That's possible. I haven't looked at that yet, but the CSR was actually not all that hard to collect once we kind of got it in the rhythm of it. Most of the difficulties were associated with trying to test the downwelling channel and if it's meaningful and trying to correct for it. And I mean, obviously there's some issues with the unit, but I think once you've done everything for the first time, it does get a lot easier to do that. I mean, you're out there for a couple hours, and also another benefit of doing the CSR is that you get a lot of data. You can so you can use a water index like we did, or you can use a vegetative index. You can look at leaf area indexes. You can look at all kinds of different things. So it is useful to have the CSR, and we think you can probably see that at an earlier time point than you can for seeing how long the grain filling period is. Which is nice, because then we might even be able to see the phenotype before the plants have sent up the spikes to start filling with grain. So, is there agreement between our water index and our yield that we saw showing difference between irrigated and non-irrigated treatments? We did see, I can put this thing back up here. So there was some agreement between the 2 different measurements. I was a little confused when we got to the yield data, because in each trial, the yield was higher in the water experiments than it was in the dry experiments, which I wasn't terribly worried about. But so there was significant differences between the varieties with regards to yield. There are significant differences due to the genotype and to the treatment. So basically we're saying that there was a difference in the yield of the 1RS, and there was a difference in the yield of the MA genotypes, and there was a difference in the watered, and there was a difference in the dry, but we lost our genotype by treatment interaction that we were seeing before. So there wasn't necessarily a significantly bigger difference in the yield between the watered and the dry in the 1RS as there was in the MA. So that doesn't really agree with what we're seeing in our CSI measurements, where we're seeing between the watered and the dry, there was really no difference for the 1RS genotypes, but for especially for the Han recombinant genotype, there was a huge difference. So this wasn't reflected with the yield. Like I said, this may have been because we had some really weird weather, and there was late rain, and I don't know if possibly whatever this phenotype is that's or whatever this gene is that's responsible for this, if it's imposing a higher penalty on the plants when they're subjected to water stress and then water comes, then then the plants would otherwise be subjected to if they didn't have that gene. So it might be they're adapting to the drought, and then when there's water, all of a sudden the plants that aren't adapted to the drought, they can really catch back up to the other ones more quickly than those that were adapted to the drought can deal with all that extra water. Okay, so there are no more questions here. Oh,
Okay.
Okay, I guess there's one more question. So did we notice any correlation between the water index with any yield? So maybe it can be used as a, well, like I was saying, we did see that there was some small correlation between what we saw with our water index, but we kind of lost that that genotype by treatment interaction that we were looking for. When we are using this index because in a previous publication, they found that it was highly associated with yield under conditions of different drought stress, and it is actually looking at the water status of the plants. So we're not sure if the rain that we got late in Davis was kind of confounding our drought tests, because there might be some interaction with adapting to the drought, and then when a bunch of rain comes, it might penalize the drought-tolerant plants more than those that aren't drought-tolerant. So there wasn't that correlation between the NWI3 and the yield components that we were looking for this year, but we have seen in the past that we've seen in the past basically with yield what we saw with NWI3 this year. So we do think there's a good correlation between the NWI3 and the yield at the end of the year, given that the weather cooperates. So as you're saying, maybe we can use this as an indicator for drought tolerance. That's exactly what we're trying to use this for. So this is when I was talking about trying to get a phenotype so that we can nail it down earlier. We are using this NWI3 index. We would like to use this as our phenotype for whether the plants are drought tolerant or not drought tolerant.
Okay, any other questions? Okay, is there, before everyone disperses, is there anyone who hasn't yet joined the Plant Breeding Training Network community? I think you can just pick Dina to verify with. Or you can pick my name. She should be on there. If you look for Dina Namath Covert.
We're also trying to work out some of the extra bugs that kind of came along with the new web page design. So I'll let Dina know that you guys are having a little bit of trouble in there, and we'll talk to the programmers.
Yep, we can walk through that quick. So, Tyson, can we, oh, never mind, there it is. Yeah, if you're having problems getting to the discussion board, and you could send an email either to Dina or to me, that would be great. Just letting us know exactly what you're running into. Probably to Dina. I'm not sure if you guys have my contact information. Okay, so walk you through here. So everyone found the instructions to get in fine? After you've created an account, you should see this little triangle, this little yellow triangle with an exclamation point in it. And that's what's going to lead you to the verification screen. And provided they haven't changed anything, you should be provided with a drop-down list of all the members of the community. So for the Plant Breeding Training Network, you should have Dina Namath's Culvert verify for you. So you select her name and click Submit. And then as soon as that little triangle goes away, you should be able to post to the discussion board. Does anyone have any other questions? Everyone does have an account. It's just the verification that we're having trouble with. Okay, if there aren't any other questions, thanks for coming.